YouTube is working. Yo, what's up, YouTube? It's your boy Bide. I tried to stream on Twitch, Kick, but it's not working as I wanted. It's interesting. So let me just where's the stream plug? Streaming disabled. Oh, I gotta go paid. Upgrade my plans. Nah, fuck that. Not gonna do that. Well, until then, Kick, I guess. Kick ain't got shit. Now, okay, then I have one more thing to do. I need to check my YouTube live stream description because I tried adding. Okay, there we go. That's good. That's there. Now I need to get this onto the gameplay video. So how do I add this? So Okay, copy, edit the whole thing, paste, and YouTube. Oh, yeah, we got him. Got him. Mm-hmm. Yep. Yup, 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 yup. Now I need to do this every day. Cause
Yo, what's up, everybody?
also I need to change the word steady. The word steady is so. It's not that. It does not ring a bell. Knowledge hunt. You know, some words like that. Hunt for knowledge. Shit like that. You we gotta do some words like that. Twitch is literally, Twitch is literally twitching. Yeah, boy. Well, that's unfortunate. I cannot stream on Kick. Or whatever. I don't care. Genetics, PDFs. It's Watson Crick Foundation. Okay. This is how old people look like. This is how they talk. To the bottom. To the left. And deep. Seek. Seek that deep. Oh yeah. Oh yeah. What the That's there. Not now. Oh no. Not now. What? It's not now. Okay. Yesterday I looked into three main foundations leading to the discovery of DNA structure. Mm, it was the double-stranded helical structure by Rosalind Franklin. Then the Chargaff's rule. Then the last one. I forgot. The Chargaff's molecular dimensions and bond lengths by Linus Pauling. One more thing. I'm so hungry, man. Oh. If I understand, I don't need this Chrome. No, I don't need that Safari tab. I'll just close it, and yeah, it's just how it is. Mm, mm. Okay, so I don't know exactly how much was the bond length and stuff, but I know it's 20 Armstrongs, something like that. So let's go past the next one. Oh no, I just skipped a lot. What is the structure of chromatin? That's the next question. Let's see what these two AI titans gotta say. Also, I need to see what the YouTuber got. Holy fucking shit! It's the Epstein files. What is chromatin? 4 minutes. I need the most. Ah. This looks good.
Het is eindelijk sale bij TUI. Tot en met maandag met korting tot wel €400 per persoon. De allerbeste last minutes boek je nu tijdelijk extra voordelen.
Just gonna fucking break this ads, bro. Oh.
All right, in this scene over here, we're going to talk about a very basic topic. The structure of chromatin. And we're going to make it really easy and a lot of fun. So let's begin. Here we see the histone octamer. The histone octamer consists of H2A, H2B, H3, and H4. Wait a minute, that's not an octamer. Those are 4 molecules. And the answer is that these are doubled. That is, there are 2 of each of these molecules over here. That's why it's called an octamer. And what happens is, DNA loops about 2 times around each of these histone octamers. And this is known as a nucleosome. Wait a minute, is that a gun? That's right, gun reminds me of 1, which reminds me of H1, as H1 is the linker molecule which connects these beads to the string. both at the entrance and the exit of the nucleosome. So there we have it, the chromatin structure.
That's
All right,
it?
I hope you enjoyed this scene
Oh, yeah.
on histone structure.
It's DNA trying to wrap themselves on this stone-like chromatin. Organizational animation. Let me see what they got.
Today's topic is chromatin organization.
So Russia.
DNA is held together by histone and non-histone chromosomal proteins, which are collectively known as chromatin.
Dude.
A human
Sup?
cell is made up of 46 chromosomes. If those chromosomes are stretched and joined end to end, they could reach up to 2 meters.
What's
But
behind
the
me?
nucleus has
Oh,
a very
it's okay, it's from their video. Okay.
space to hold such long
Whoa,
chromosomes.
whoa, whoa! I did not see.
Joined end to end, they could reach up to 2 meters. But the nucleus has a very small space to hold such long chromosomes. So, it is obvious that chromatins needs to be highly condensed in order to be placed inside the nucleus.
Yeah.
Chromatin appears as a series of 11 nanometer beads which are wrapped by DNA of approximately 200 nucleotides. Each bead with associated DNA molecule is known as nucleosome, the unit of chromatin. The nucleosomes on the DNA chain is called beads on a string structure.
It's in a string structure.
If
That's it.
one nucleosome core particle is subjected to dissociation with increasing salt concentration, we will get a octameric histone core protein and a 147 base pairs double-stranded DNA. This
147.
147 base pairs long DNA wrap 1.7 times around the histone core proteins. The octameric structure of core histones were suggested by Roger Kornberg in 1974.
Kornberg.
H2A,
Oh,
H2B,
come on, bro. You're
H3,
pushing this.
and H4. These are 4 types of histone proteins, each of which is present in pair to form the octameric structure of the histone core.
Let me see.
It shows the 3D conformation of the nucleosome core particles. Each of the core histone protein contains an N-terminal tail and a histone fold region. All 8 N-terminal tails protrude from the core structure. They provide binding site for several other proteins. Every nucleosome has 142 hydrogen bonds between the DNA and the histone core. The AT-rich minor groove of DNA binds with the arginine and lysine residues on the histone core proteins. These amino acids are positively charged so that they can tightly bind to the negatively charged DNA. The outer side of the minor groove of DNA is GC-rich. Another important factor is an additional histone that is often present in a 1:1 ratio with nucleosome cores known as H1. Each H1, which is also called linker histone, binds to each nucleosome, contacts both DNA and protein, and changes the path of DNA as it exits from the nucleosome. Nucleosomes are arranged in a complex zigzag structure known as 30 nanometer chromatin fiber. These fibers further form 300 nanometer loops. This is the general overview of chromatin organization.
chromatin organization. I still didn't get it in a full form, but yeah, this is how you start.
My work is easy because I use ClickUp. Here's how I keep track of all my tasks. I start by brain dumping everything I need to get done.
The human genome contains over 3 billion base pairs of genetic information. If it were stretched out end to end, it would span approximately 6 feet. But each human cell that houses all this information
No,
is
I
about
6
0.0002
feet. I saw the last video was like if you stretch your DNA, it's gonna go past the sun 2 times from Earth. That's what? 6 feet?
But each human cell that houses all this information is about 0.3 billion base pairs of genetic information. If it were stretched out end to end, it would span approximately 6 feet. But each
Human genome stretched out. I need to fact check this because you gotta think on that. Six feet. I believe 6 feet is. Okay, 6 feet. I'm not 6 feet, but yeah, I get it. So I saw. Okay. Okay. I need to fact check this. What did I hear last time? Let me just. DNA structure and replication.
Molecule, but rather a pair of molecules that are held tightly. Assume that you are a human, in which case, complicated molecule that exists, and potentially the most important one. Sure, what it is that there are probably a body.
Okay, stretch the
I
sun.
consider that there are probably 50 trillion cells in my body right now. Laid end to end, the DNA in those cells would stretch to
Okay.
cell that houses all this information is about 0.0002 inches across, approximately 360,000 times smaller than the length of all of our genetic information laid out side by side. So how in the world are we able to fit that massive amount of information into a tiny cell? As always, nature came up with a clever solution. Instead of all that DNA being stuffed into cells as long, sinuous strands, which wouldn't leave room for much else and would be hard to untangle when the information is needed, the information is coiled up around a series of proteins into a highly organized, condensed structure called chromatin. As you'll see, it's similar to the process of turning wool into a knitted sweater, where each piece provides structure and organization to the next. Let's explore this concept a bit more. First,
That's good.
a small segment of the DNA double helix is wrapped around a collection of proteins called histones, which forms a structure called a nucleosome. Nucleosomes then fold on top of each other into a single chromatin fiber. From here, the chromatin fibers are further coiled and intertwined into a chromosome, which organizes our genetic information in the form of genes, units of DNA that provide instructions to make proteins. Each human cell has 23 pairs of chromosomes, or 46 total, which contain approximately 20,000 to 25,000 genes, all of which live inside the cell's nucleus. From here, all that neatly packaged information is responsible for making sure each cell, and in turn our whole body, functions properly. It turns out that this packaging trick doesn't just save space inside a cell, but also makes sure DNA is able to do its job properly. Think of it like a coiled up elastic band. When it is compacted together, no individual strands are accessible, but you can pull away particular pieces of the coiled band as needed. Similarly, when DNA is tightly wound around histones, also called heterochromatin, it isn't accessible to the proteins required to read DNA, so those genes remain turned off. In response to environmental changes, however, histones are altered through a variety of molecular processes, which triggers the DNA to uncoil into a more relaxed state, known as euchromatin. Euchromatin is accessible to DNA-reading proteins, thus turning the necessary gene on. These dynamic changes in chromatin density, a process called chromatin remodeling, are important for the process of cell division, when cells copy all of their genetic material and then segregate and repackage it into the newly formed daughter cell. In non-dividing cells, chromatin remodeling allows for tighter control over when and where certain genes are activated, which ensures complex organisms like humans run like a well-oiled machine. Beyond ensuring the right DNA is accessible at the right time, this packing structure also prevents DNA from getting tangled, which reduces DNA damage. The structure of chromatin is one of biology's many ingenious solutions to a mathematical challenge and simultaneously solves numerous cellular complexities. Number one, it ensures vast amounts of genetic information can fit into each cell. Number two, it enables complex organisms to diversify their functionality. And number three, it helps prevent DNA damage. More levels of biological intricacy, however, also means more opportunities for mistakes to happen. Researchers are finding that genetic mutations affecting chromatin structure are common in many types of diseases, especially cancer. While scientists are still trying to uncover the precise roles these mutations play in diseases, efforts to develop drugs that target chromatin machinery have shown promise in early clinical studies, revealing their potential to provide a new type of medicine for challenging health problems.
Interesting to say the least. Okay. So, what is the structure of chromatins? Chromatins are spherical proteins. Chromatin is a complex of DNA that proteins that make up chromosomes within the nucleus of a eukaryotic cell. Its structure allows long DNA molecules to be compacted and functionally regulated. The organization of chromatin is hierarchical and dynamic, adapting to cellular needs. What do you mean hierarchical? How is it hierarchical? Hierarchical. Hierarchical. Nucleosomes, the basic unit, core histones, chromatin consist of repeating units called nucleosomes, each composed of a histamine octamer, H2A, H2B, H3, and H4. 147 base pairs of DNA wrapped and around the histone octamer in 1.65 turns. The repeating units called nucleosomes, each composed of histone octamer, 2 copies of 4 core histones, 147 base pairs of DNA wrapped around histone octamer in 1.65 turns. Linked DNA connects nucleosomes, typically 22, and is associated with linker histone H1, which stabilizes the chromatin fiber. High order structure, 10 nanometer fiber bead on a string. Nucleosomes connected by linker DNA, visible under electron microscopy. 30 nanometer fiber, a more compact helical structure by nucleosomes arrays folding onto a solenoid. I forgot the solenoid. Okay. This structure is less consistently observed in vivo. The fuck is a vivo? Chromatin loops, the 30 nanometer fiber further folds into loops. Anchored by cohesion and CTCF proteins forming topologically associating domains. I don't know what these are. Chromosome territories. Chromatin loops condense further into distinct chromosome territories within the nucleus during interphase. During mitosis, chromatin undergoes maximal condensation to form metaphase chromosome. Chromatin states and modifications. Oichromatin. The fuck are these names? Oichromatin, euchromatin. It's not oi, it's euchromatin. Euchromatin, less condensed, transcriptionally active, enriched in acetylated histones, H3K4Me3 marks. Heterochromatin, highly condensed, transcriptionally silent. Okay, this is the one that is used for transcription. These are transcriptionally silent. Enriched in H3K9Me3 marks and H3K27Me3 marks. Facultative heterochromatin, silenced temporally. Example X chromosome inactivation. Constitutive heterochromatin, permanently silent. Example centromeres, telomeres. Key proteins and factors: histone modifications, acetylation, methylation, phosphorylation, regulate chromatin accessibility, chromatin modelers, example SWI, SNF, ISWI, reposition nucleosomes, non-histone proteins, HP1 polycomp group proteins, maintain condensed states, functional significance, DNA compaction, fits 2 meters of DNA into a nucleus, 10 nanometer diameter. Gene regulation, chromatin structures controls transcription, factor access, DNA replication and repair. Chromatin dynamics facilitate these processes. Dynamic multi-level structure that balances DNA compaction with the regulated gene expression. What do we got? Chromatin is a complex DNA and protein found in the nucleus of eukaryotic cells. Its job is to package the long strands of DNA so they fit inside the nucleus and they can be regulated. Okay, it's not just packing, it has the regulation also as a factor. Okay. Basic structure: DNA plus histamine proteins equals nucleosomes. DNA wraps around 8 cores of histamine protein called histone octamer. About 147 base pairs of DNA wraps around each core. Looks like bead on strings. Under microscope, each bead equals a nucleosome. Linker DNA: short stretches of DNA between nucleosomes like a string between beads. That's the linker DNA. 30 nanometer fiber, solenoid, or zigzag model. Nucleosomes coil into more compact fiber, 30 nanometer thick. Is it the thickness of the nucleosomes or the fiber they are forming? With the help of H1 histone. This is more condensed than beads on a string. High order folding. 30 nanometer fiber folds further into looped domains. These are called chromosome arms. During cell division, chromatin condenses even more to form visible chromosomes. Chromatin is composed of DNA tightly wrapped around histone proteins to form nucleosomes, which coil into 30 nanometer fiber and further fold into high order structure, ultimately forming condensed chromosomes during cell division. Let me know if you want. Nah, I don't want. Okay. Telomere, yeah. Okay. Linker DNA. Interesting. Well, let me just finish all these questions first. Ozaki fragments. These are internal DNA stuff. Lagging strand. The biotech.
De veilige huis van... Nu met gratis plaatsing van een videodeurbel. Ga naar verysure.nl.
These are old.
When DNA replicates, Its strands are separated by the enzyme helicase. Single-stranded DNA binding proteins keep the strands from re-annealing. One DNA strand encodes the leading strand, which forms from its 5' to its 3' end, using DNA polymerase III. No problem here, but the lagging strand presents problems. It has to form from 5' to 3' too. It forms in pieces called Okazaki fragments. First, an RNA primase lays down an RNA primer. Then, DNA polymerase III lays down new DNA.
Opposite direction.
The process repeats again and again. DNA polymerase I replaces the RNA primers with DNA. Finally, DNA ligase links the Okazaki fragment.
Okay, the Okazaki fragments are like final touch.
In this video, we're going to focus on DNA replication. The first thing that you need to know is that DNA replication is semi-conservative. What does that mean? So looking at the strand on the left, that is the original strand. You can call it the old strand. And on the right, it was replicated. We have two new strands. In each new copy, we could see there's one red strand and one blue strand. So we have one old strand, that's the red one, and one new one, that's the blue one. So thus DNA replication is semi-conservative. Each new strand retains one original copy, and it has one new copy. Number two, the two DNA strands are anti-parallel. This means that they run in opposite directions. So from left to right, one of the strands run in the 5 to 3 direction, and the other one goes in the 3 to 5 direction from left to right. Now, the DNA strands are also complementary. They have complementary base pairing. So for instance, let's say that the nucleotide sequence of the first strand is A, T, T, G,
Oh shit,
T, C.
TAA.
If we know the sequence of one strand,
CTA.
we can predict the sequence of the other strand. Now, in DNA, you need to know that A always pair up with T, and G always pair up with C, and vice versa. So here we have A is going to pair up with T is going to pair up with A, G is going to pair up with C, A is going to pair up with T, and C will pair up with G. So given the information of one strand, we can write the nucleotide sequence of another strand. And so that's the idea of complementary base pairing in DNA. Now the next thing you need to know are what holds nucleotides together. And the answer is hydrogen bonds. Between adenine and thymine, there are two hydrogen bonds.
Guanine and cytosine.
And you need to know that
three.
between guanine and cytosine,
Cytosine,
there
yeah,
are 3
whatever.
hydrogen bonds holding them together. So those H bonds are holding the DNA strands together. Now, there's a lot of other stuff that we need to talk about. One of which is that DNA replication usually proceeds bidirectionally. So what does that mean? Well, let me draw a picture that will illustrate this concept. So this bubble that you see between the two strands, that is the origin of replication. In red, this is a new strand being produced. Now, in bidirectional DNA replication, the strands can be produced in both directions. Bi means two. Now, another example of replication is unidirectional replication. So that occurs when the strand is being synthesized in one direction. So here's a picture of the replication fork that I decided to draw ahead of time. And the first thing that we're going to talk about is the helicase enzyme. Helicase separates
The
the
hydrogen
two
bond.
strands.
Yeah.
And it does so by breaking the hydrogen bonds that hold the nucleotides together. As helicase separates the two strands, torsional strain is created ahead of the replication fork. And as a result, an enzyme is needed, DNA gyrase, which is a type of topoisomerase. And these types of enzymes, they can reduce the torsional strain and even relieve positive supercoiling that could form in the DNA strand ahead of the replication fork. So I'm going to put T for a topoisomerase enzyme. So you might see DNA gyrase here as well, but keep in mind that DNA gyrase is a type of topoisomerase enzyme. Next, we have the SSB proteins, the single-stranded binding proteins. These SSB proteins, they protect the two strands from cleavage. They also stabilize the two strands, preventing them from snapping back together. Now,
It's a
DNA
protein,
replication
okay.
requires an RNA primer to begin. So I'm going to draw the RNA primer in green. The RNA primer is basically a sequence of RNA nucleotides. DNA polymerase 3, once it sees the RNA primer, it begins to synthesize the new strand in the 5 to 3 direction. This strand here is called the template strand. And that strand runs in the 3 to 5 direction. But DNA
Just a minute.
DNA polymerase 3 is going to build a
Oh
new
shit,
strand
my neck.
in the 5 to 3 direction. So make sure you understand that. DNA polymerase 3 adds nucleotides in the 5 to 3 direction. Now, the enzyme that creates the primer is known as primase. Now, DNA replication is semi-discontinuous. Now, what does that mean? What does it mean that DNA replication is semi-discontinuous? Well, we need to talk about the other strand. So the first template strand on top runs in a 3 to 5 direction, which means the template strand on the bottom. So once a primer is added to this strand, DNA polymerase 3 is going to work in this direction. That is, in the 5 to 3 direction. Now, notice that
Oh.
this strand, by the way, is called, the one in red, this is called the leading strand. And this one here is called the lagging strand. The leading strand moves in the same direction as the replication fork. The lagging strand moves in the opposite direction of the replication fork. So as the replication fork continues to move forward, you're going to have more space here. So what's going to happen is a new RNA primer is going to join here, and then DNA polymerase is going to start there, producing or adding nucleotides in this direction. So in the leading strand, DNA is, so what's going to happen is a new RNA primer is going to join here, and then DNA polymerase is said to be semi-discontinuous. It's continuous in the leading strand, but discontinuous in the lagging strand. This part, this RNA primer, gets removed, and so I'm
Okataki. Okataki. It's Oz. Oh.
polymerase 1 and 3, they have 3 to 5 exonucleotide and replace it with the right one. So that would be the proofreading ability of DNA polymerase 3. Now, DNA polymerase 1, not 3, has and then also DNA polymerase 3.
First let me just know there's no fucking paste. There's no fucking paste. This guy's still writing. Sketch how translation proceeds at ribosome and label main important components. What is a chaperone? Name at least one. Yeah, I get it. But just by learning these questions, it just just by learning these questions, it does not just.
Okay, so the topic of this short video today is going to be the 3 different types of RNA: mRNA, tRNA, rRNA. So let's go ahead and get started. Okay, so let's talk about ribonucleic acid. When you look at the picture, RNA on the right, you can see it's only a single strand of nucleotides. But the picture of DNA on the left, DNA is a double helix because it's made from 2 strands of nucleotides. Well, RNA is still a nucleic acid, so it's still made from building blocks called nucleotides. And nucleotides have 3 parts, one being a phosphate, a second being a sugar molecule. Now, the picture that just showed up, this is the sugar called ribose. This is the sugar of RNA. On the left, this is the sugar called deoxyribose. This is the sugar of DNA. They're virtually identical. Highlighted in red is the difference. The sugar on the left, deoxyribose, it's been deoxied. It's missing an oxygen atom. Other than that, they're very similar to one another. And then the third part of a nucleotide is the nitrogen base. In RNA, the nitrogen base might be A for adenine, it might be G for guanine, it might be C for cytosine, and it might be U for uracil. These are the 4 nucleotide nitrogen bases.
Instead of thymine, it's uracil.
And ultimately, we're going to learn about the 3 kinds of RNA. We're going to learn about mRNA, and then we'll learn about tRNA, and then we'll learn about rRNA. So let's talk about mRNA first.
Let
Okay,
me
so the
just
of mRNA stands for messenger, messenger RNA. And you're going to see it's going to start in the nucleus of a cell, but it will end up at a ribosome. Well, here's a cell right here. And let's talk about eukaryotic cells. First, eukaryotic cells have a nucleus, and in that nucleus is the DNA of the cell. Now, you might know that DNA is the cell, but it will end up at a ribosome. Well, here's a cell right here. And let's talk about eukaryotic cells. First, eukaryotic cells have a nucleus. A ribosome is the actual builder of the protein, and that's out in the cytoplasm. So what does mRNA do? It makes a copy of the DNA code. It makes a copy of the DNA sequence. It makes a copy of the DNA instructions. And messenger RNA will deliver the instructions from the nucleus to the ribosome. And once the ribosome has the instructions,
whoa.
what the ribosome will do is will build a protein. And it does that by gathering up a bunch of amino acids and linking them in a long chain, which eventually is protein. And this is a process called translation. We're going to learn more about translation in another video, but that's what a ribosome will eventually do, is gather the amino acids and link them into a protein. Okay, and now for transfer RNA. The T stands for transfer RNA. And the location is going to be in the cytoplasm. Now, in my cell drawing here, I removed the nucleus from the drawing just to get rid of some of the clutter in the drawing. But this is also in eukaryotic cells. And what happens is, we said a moment ago, the ribosome is going to gather amino acids to make a protein. But how? How does the ribosome actually obtain the amino acids? Well, transfer RNA will pick them up and the ribosome, and one by one, the ribosome, and one by one, the ribosome, and one by one are RNA. And so
What happened to it? What was that? Yeah. It's 3:30. Oh shit, it's still not. It's 3:30 right now, and I gotta cook my beef. I cooked some beef up here. So okay. So I'm gonna cancel the live stream. The Twitch stream is gonna be continuing, but YouTube stream, peace out, YouTube. I'll be